into about 15 different organizations. We're the most southern of this, the most southern organization. And what we're trying to do is basically restore the IRL from the northern to the southern end. And so different organizations are doing different restoration projects. So some people are focused on mangroves and some people are focused on oysters. And our work is focused on 3 different things. One is clams and seagrass, which is sort of a recurring theme here. And we also were asked to put some artificial reef structures under our deck in the back to ask how they bring in different organisms. And so Evelyn has been working on characterizing how those reef structures have changed over time.
So my name is Evelyn Marquez, as Dr. McCoy said, and I will be presenting on ecological succession using artificial reefs in the Indian River Lagoon. So let's start off with ecological succession, which is how a community changes over time. This could be species coming in an environment or leaving. It is important because it helps scientists understand and predict ecosystem recovery. This is really important in habitats that have been degraded over time. The Indian River Lagoon is a 156-mile-long estuary that stretches on the east coast of Florida, stretches north past Orlando and as south as Hope Sound. What makes it really special is it's where oceans and river inlets mix, which creates brackish water. Brackish water allows 6,000 different species of plants and animals to live in this lagoon. However, over the past several decades, the Indian River Lagoon had a huge loss in its biodiversity. The destruction of Coquino Rock. The Indian River Lagoon has been negatively affected by many things, as stated in the last presentation by Nick with the seagrass. There was this hard limestone structure, which is pictured right here. It has shell fragments in it. This was once the hard structure that was in the Indian River Lagoon all along it. However, as the Indian River Lagoon started getting developments and restaurants built along the coast, humans have taken out this rock, which was the hard structure that was once in the IRL. And this was important because it was a habitat. So why are artificial reefs important? Artificial reefs are man-made structures that imitate real reefs. These can be used as sunken ships or just concrete, and they provide a habitat and create an ecosystem for animals. This brings me to my research question is, what is the natural succession pattern on an artificial reef across multi-trophic communities in the Indian River Lagoon? The knowledge gap is there has been limited research that has examined how artificial reefs support ecological succession in the Indian River Lagoon. This study evaluates whether artificial reef structures promote organism settlement and community development. And this work investigates the potential of artificial reefs for habitat restoration. The experimental design is we use the Spartan artificial reef, which are X-shaped concrete reefs. A total of 36 were placed under the back of dock property into the Indian River Lagoon. Looking at the schematic of the reefs under a dock, there's a total of 36, so we made them 4 groups of 9, A, B, C, D. Looking at D, you can see it's more closer to shore this is when you're walking onto the dock, and A, B, C is on the dock, more into the water. We split them up into 2 groups because for some of the samples, we would randomly use Google Generator to pick us 2 numbers from each group to give when we did samples to always keep it randomized. The first objective is to examine the microbial abundance. The methods used is we would swab the top, the north, and the south side of the reef, inoculate it on a marine agar using an isolation streak, incubate the plate for 48 hours, and record the colony abundance by hand counting it in the lab. The unit used for this was a colony forming unit. What is a colony forming unit, you may ask? It is the circles that appear on the plate Looking at those, you can visually see that those are one or more living microorganism in that dot. And they're easy to hand count. This is the unit we used. So for the first 4 weeks, we took swabs on selected reefs. And after incubation, I would hand count the colony forming units that were on each of the plates. Out of the 4 weeks, we created this graph. The x-axis is the different weeks. January 16th was the date of employment, and the y-axis is the average of colony forming units. Seeing that the day that we deployed the reefs, they were microbials on the reefs already on date of employment, and throughout the past 4 weeks, they increased each and every week. The second objective is to quantify the fouling species coverage. Fouling species were quantified by estimating the percent coverage across the surfaces of the reef. Month 1 was when we can visually start seeing things growing on the reef, as green algae was seen growing on the surfaces of the reef. The second month is when different species started settling on the different surfaces, creating a fouling species community onto the reefs. Month 3, we continued to see an increase in different species, as well as continuing seeing an increase month 4. Out of all the months, we created this chart, which the x-axis is March, April, May, June. And the different colors represent the different species that were growing on the surfaces of the reef. As in March, you can see the red. That shows algae, which was seen a lot in March and April. However, mid-April, you can start seeing many different colors, which represents that there's different organisms growing on the reef. And throughout April, May, you can see the green, which increased a lot. That is known as biofilm, which is a mixture of bacteria and mud that would harden on the reef surfaces. We also created different graphs from the different sides. The top is the north, the middle is the south, and the bottom is the top. Looking at the top, we mainly just saw algae and biofilm growing on it, but the north and south is where we really saw different organisms settling on it and a community of fouling species on these surfaces. The third objective was to assess the invertebrate diversity. For this method, we use biobox. What is a biobox, you may ask? They're commonly used in experiments. They are black plastic trays that I would cut window screening around the perimeter and zip tie it around it. We would put 1 bag of oyster shells per biobox. This helps invertebrates such as crabs want to colonate inside of it. They colonate inside of it so they can hide from predators. These were placed around the reefs randomly to see the invertebrate community around the reef. After 3 months, we pulled up bioboxes. We pulled up one at a time, dumped it onto a water tray, and put the individual species in a bucket they belong, and then would count the different species that were in the bioboxes. This is a pie chart showing all of the different species in all of the bioboxes. This is the furrowed mud crab, which was most seen. 40% of the species is in the different bioboxes. As the green right here is a porcelain crab. Here is the biobox species richness, showing the different species shown in each of the bioboxes, as well as each biobox is pointing to the different species that are inside of it. An observation I did make is that in biobox 4 and 15, which are closer to shore, they were seen to have not as much as a diversity in biobox 68 and 97. Those were seen to have a huge increase in different organisms. As we're going to continue doing this experiment and see if this is a pattern that continues, but this is an observation I did make looking at the data. The last objective was to identify the different fish species. GoPros were used to capture the organisms swimming around. As we used a PVC pipe, we would put the GoPro on it and it was anchored around the dock. This would record once a month. We would record an hour in the morning and an hour in the evening to see fish activity around the reef. As well as we did fish survey. Two people would start at separate ends of the dock, and we would swim around the dock recording the fish utilizing the reefs as well as the fish around the reefs. Over time, on February 20th, using GoPro films, I promised there's a sheep's head swimming under the reef. He was using it for habitat. 4 months later, on June 17th, you can see a huge increase in the fish. These are mangrove snappers, showing that over time that the fish species did increase, as I assume they are attracted to the things growing onto the reef, forming a community. So from January to June, the species diversity and abundance did increase, creating a community underneath the dock, supporting the idea that artificial reefs is an effective tool for habitat restoration. For future analysis, ecological succession is a process that takes time to build a community, sometimes even decades. This is an ongoing project I'll be working on for another year, such as doing fish survey once a month, pulling up bio boxes every 3 months, and recording the fouling species once a month. I would like to thank NOAA for funding my project. I would like to thank Charlie and Beth Sumberler for the suppliers. A huge thank you from Kalita from One Lagoon for help giving me this internship, and a huge thank you to my mentors, Dr. Kyle Bartow, Dr. Krista McCoy, Nathaniel Wynn, and Nicholas Curto. Any questions?